Not ordinary, no, it's something new. You could be looking, but you haven't seen this before. Unreplicated, it's not deja vu. The one and only. I'm one of a kind. Tip top of the line. I'm one of a kind. Tip top of the line. I'm one of a kind. Contemporary or original, nothing like it, never seen it like this before. Authenticated, no identical. There's one and only. I'm one of a kind. I'm one of a kind. Not ordinary, no, it's something new. You could be looking, but you haven't seen this before. Unreplicated, it's not deja vu. The one and only. I'm one of a kind. Tip top of the line.
Hi, warm welcome to all of our viewers and listeners to Ask the Expert, and especially to our guests, whom I will introduce shortly. My name is Sean McGrath, and I'm the Medical Strategy Lead for the My Name's Doddy Foundation. Those close to the foundation will know we do a regular Dodcast, but more recently we have introduced Ask the Expert session for two reasons. Firstly, your feedback, which we take very seriously, and also Doddy always felt that he was very lucky. Because of the foundation in his name, he had access to many experts, and he felt that wasn't fair for everyone else. So he really made sure that we always will facilitate access to those very same experts. So today we're talking about clinical trials in motor neuron disease, and I'm delighted to introduce two special people. Professor Dame Pam Shaw, the director of CITRAN, which is the Sheffield Institute for Translational Neuroscience, and truly a global leader in MND research, and Dr. Rob Wainwright, who captains Scotland Rugby, of course a close personal friend of Doddy's, and the driving force behind Doddy Aid. Doddy Aid, of course, has been very visible over the first 6 weeks of the year, and in fact every year now. And thank you to Rob and all those who participated by running, walking, swimming, etc., cycling. Thank you to everyone. So Pam and Rob, you're both extremely welcome. Can I ask viewers to keep sending in questions via the comments page on Twitter, Facebook, and of course, YouTube. Any questions we don't get to, we'll make sure that we post onto the website later. So, Pam, over to you for the first question. I'd like you to paint a picture, if you will, of the clinical trial setup in the UK or the landscape before the Doddy Foundation was an entity, and compare that what it was like to what it is like now.
So I think the landscape has changed enormously over the last couple of years, really. So in the past, there would be an occasional pharmaceutical company that would want to test a particular therapy in MND. We tended to have one trial at a time. Academic-led trials were very rare, and the number of MND patients that could participate in those trials was very small and limited. I think there have been two recent trials of one called Miracals, one called Valor, that we've learned an awful lot from and create a real ray of hope for the future of trials. But I think also we're moving towards now trying to test drugs that are known to be safe in human beings and that already have a license for use. And with the sophisticated models that we now have, we can test those drugs in cell lines derived from the skin cells of patients and identify molecules that may have protective effects for the motor neurons. So there's a real development now of repurposing or analyzing whether existing drugs could actually have another indication. And there are some trials where that is already happening. MND Smart and the Trust trial are examples of that approach. I think the other thing that has changed is our ability to monitor whether a treatment is doing any good has improved as well, with the identification of biomarkers, which we'll perhaps talk about a little bit later. But biomarkers are items, molecules, chemicals that you can measure in blood or cerebrospinal fluid that tell you whether your experimental treatment is hitting the right target, so getting to the right place, hitting the right target, and lowering the injury to motor neurons. So I think, since the My Name's Doddy Foundation was established, there have been real changes in what is happening in relation to clinical trials, and the support from the foundation has really enabled that to happen. So more than 400 patients now are taking part in MND Smart. So one of the changes in trials that we want to develop is making experimental treatments in trials more available to more patients. In the past, the inclusion and exclusion criteria have been quite restrictive. So we're on a mission to make trials more accessible to more of our patients.
That's fantastic. And tell me about there's two trials that spring to mind that have reported some data fairly recently. Tofersen is one of them, and the interleukin-2 trial from Myricals. You mentioned those very briefly. Can you just give us a top line on those two and what they might mean for patients in the future?
So Tofersen is a genetic therapy trial aimed at patients who have a change in the first gene that was found to cause familial MND. So that's where MND is affecting more than one member of a family. The gene is called SOD1, and we did a lot of work, including in Citran, in preclinical models, so in cell models, in mouse models, in fruit fly models, showing that if you lower the level of the SOD1 protein within the nervous system, you slow down the disease course quite dramatically. And all that body of preclinical science gave confidence that a similar approach might work in human beings. So we sponsored by Biogen, the pharmaceutical company based in Boston, we did phase 1, 2, and 3 trials. And the treatment is given via lumbar puncture into the spinal fluid, and the ASO lowers the level of the SOD1 protein, and the results are slightly complicated, but overall really very positive. And I've taken part in more than 23 clinical trials for MND. This is the first trial where patients have been sending videos of themselves doing things that they couldn't do a few months ago. So writing my Christmas cards this year, I couldn't last year because my right hand was so weak, climbing up the garden steps, which I haven't been able to do for 2 years. So wow. A lot of patients reporting stabilization of their condition, but some actually noticing real improvement in their muscle strength. And the other very positive thing that emerged from the Tofersen trial, so we could measure the lowering of the SOD1 protein in the spinal fluid, but we could also measure a lowering of neurofilament levels. And neurofilaments are proteins that are present in high abundance in the motor neurons, and they get released into the spinal fluid and blood as motor neuron injury and cell death is happening. And Tofersen actually lowered the levels of that biochemical marker of motor neuron injury, so convincing us that we were really doing something good. But what we also learned from that trial is that the clinical effects, so we could measure these biochemical markers at 3 months, 6 months, but the clinical effects took longer. So at 12 months of follow-up, the clinical benefits were very clear. So I think what we've learned is if we've got a treatment that's working, it still takes time for motor neurons to heal and to start resupplying the muscle fibers with nerve fibers or electricity, if you like. And I think, you know, we could learn from poliomyelitis. People could be really weak from polio, but once the damaging virus had gone, they could make remarkable recoveries, but it would take months. It isn't an instant thing. So I think we've learned that we've got to let the nervous system heal itself over time. The second trial, Myricals, is a trial of an injection treatment. So you give an injection into the skin, a bit like somebody with diabetes would inject their insulin, for 5 days every month. And what low-dose interleukin-2 is aiming to do is increase a particular type of white blood cell in the blood called Tregs, or regulatory T cells. And earlier work from the US showed that just following up a large group of MND patients, those that had a high Treg count had a much slower and more gentle disease course compared to people that had a lower Treg count. So what we were doing in my accounts was increasing the Treg count. And again, the results need studying in some detail, but particularly in patients that have only mild or moderate speeds of disease progression, there seemed to be a huge benefit, a 76% improvement in life expectancy over the 21 months of the trial.
Wow.
So very, very exciting. And again, we've learned a lot, I think, about how to do trials better in the future from those two recent trials.
Thanks, Pam. That's really, really helpful and really exciting data that's coming through. And this is all new, and I think everyone, all the people are really excited about what we're learning and also some of these early results. But of course, big clinical trials like this cost money, and our foundation is aiming to help fund these trials, of course. And one of the ways we do that is through Doddy Aid, which has become an annual event. And Rob Online is the founder, really, the driving force behind Doddy Aid. Rob, can you tell us how it went this year, and what did you get up to with all your tricks and fundraising escapades up and down the country?
We had an amazing Doddy Aid, and lovely to come here and talk about what it's all about, and meet Pam again, who I had the pleasure of meeting down at a scientific conference. I see this is called Ask the Expert. I do feel a bit of a fraud being introduced as a doctor by you, Sean, which I've not been for many, many years. But it's, yeah, I'm a token non-expert among you experts, certainly. But Doddy Aid was fantastic this year. Obviously, a huge amount of momentum. A lot of positive momentum for a very negative thing, which obviously is the loss of Doddy himself, and to the tune of 38,000 people taking part. And
wow,
of those, the sign up from those 38,000 people basically made about £800,000, and then fundraising on during Doddy Aid and during the ride with the ball from Cardiff up to Murrayfield added another £1.2 million. And it's staggering figures, which we'd never have dreamt of a while back. And I often get asked, what happens to that money? Well, the first, I think, really important thing to say is that My Name's Doddy Foundation is extremely efficient charity, in that most of the money is going to the end cause. Their administrative costs are minimal. And in terms of the cost, this is one of the only costs really of Doddy Aid is getting snoods out to people. And I also, just mentioning it because it's been brought up, there's been a lot of misinformation about how much money Just Giving, obviously £1.2 million of the fundraising went through Just Giving, and there were scare stories about 15% being taken, and which I was actually taken in by myself. I was convinced that 15% of what was being done. But I've checked on this, and it is 1.9%, which is a far more reasonable amount of money taken off by Just Giving. So, the vast majority, well over 3/4 of that money raised, is going to the endpoint, and the endpoint is research. So, I'm not here as an expert, but I am representing the 38,000 people that were basically giving their time, their money, making the effort in order to fund research. And I suppose it's only fair that I get the opportunity to, on their behalf, to ask a question. I'm particularly interested, Pam, in drug repurposing trials, which are a wonderful shortcut in that these are drugs, I believe, I'm sure you will confirm, that have already been through all due diligence. They've been checked that they're safe, but they've been found to be ineffective in treating the things that they were being developed for, and just sit there in a theoretical pile waiting to be tapped into. And have we got trials coming up, Pam, or are there trials already working with drug repurposing?
We have, and I think one of the facets of science that has allowed that to happen is reprogramming skin cells. So patients will often give us a little sliver of skin, and we can convert those skin cells in the laboratory, in the scientific laboratory, into motor neurons or into the glial cells that are neighborhood cells of the motor neurons in the nervous system. So we can look at what the damaging factors are and then screen libraries of drugs known to be safe in human beings to see if any of them protect the motor neurons from injury. So I think it's really exciting. In our laboratories in Citran, we've got a library of 520 FDA approved drugs that we know will get into the nervous system. So neurological conditions are a bit more difficult because we have this blood-brain barrier. So the brain protects itself by keeping things out. But these more than 500 drugs we know will cross the blood-brain barrier and therefore can get to the place where the motor neuron injury is happening. So there are trials ongoing with repurposed drugs. MND Smart is one. The TRUST trial being led from Oxford is another, and there are more coming along. So we're just developing another earlier phase platform to put repurposed drugs in. And, to give one example from another disease area, we screened this library of drugs in skin cells from patients with Parkinson's disease. We found a drug that had been used in liver disease really protected the mitochondria, the batteries within the cells, which are known to go wrong in Parkinson's. And that went straight into a phase 2 clinical trial. Results were really positive. So they're now just gearing up to do the next stage. So I think that's something that's changed enormously in recent years. If you're a skin doctor or a liver doctor, you can take a piece of the damaged tissue and study it in the laboratory. We can't do that with the nervous system. We can't take pieces of a person's brain or spinal cord, but now we can recreate the cells that go wrong in the laboratory and use those model systems for screening for protective agents. So I think it is a very exciting phase.
And does this then basically remove, the lab rat part of what used to happen in the animal experimentation isn't required? You go straight from creating the cells from their own tissues and then moving straight into clinical trials, do you?
Well, it depends on what drug you're thinking about as a protector of motor neurons. There are new molecules, and we have several of them, so do other labs around the country. We have some new molecules coming through, and if they've not been used in human beings before, we have to do the animal studies and the toxicology to make sure there are no unexpected side effects. But the beauty is we've now got both models running, which should increase our therapeutic pipeline coming through into experimental clinical trials.
So Pam, could I ask you just to explain what MND Smart is, please? And we know it's out of Edinburgh. It started in Edinburgh, but it's rapidly gone over the UK. And can you just outline the concept of doing a platform trial and why it's important?
So I think they are using repurposed drugs. The literature is searched to find drugs known to be safe in human beings that might have neuroprotective properties. There is some investigation of the neuroprotective properties in the cell models and so on, and then so that they try and test at least 2 different potential neuroprotective drugs at once. When we test new drugs, we have to have a placebo group. And what a placebo group is, the patients are taking a drug that looks similar to the active treatment, but it's a harmless, innocuous substance. And so what we're trying to do is reduce the number of people that go into that placebo group by having one common group and testing several neuroprotective drugs at a time. The results are monitored live so that, and there are go, no-go points, and we've got some very effective ways of telling at an early stage whether a drug is doing some good. So if a drug looks promising, then it goes through to the next phase. If a drug after a few months is showing no evidence of benefit, then that part is stopped and a new drug is brought in. I think the other very positive thing about MND Smart is while the scientific results will be tested on patients early on in the MND disease course, it's open. It doesn't have these strict inclusion criteria, so other people can take part in the trial, for example, who've had their MND for a much longer period. Many of the pharma clinical trials are trying to catch patients very early after symptom onset, but the beauty of MND Smart is that it's open to all comers, although the rigorous scientific results will be tested in the group that are fairly early on in the disease course. So it's one thing patients have been really crying out for, access to clinical trials, and what we're trying to do is open that up so that more people can try new treatments.
Thank you, Pam. And we've had a few questions around, is it possible, is it wise to look at pre-symptomatic gene carriers or gene mutation carriers, so people that don't have any symptoms, but we screen a population, and if they have one of these genes that might be mutating in this disease, is it wise to do? Should we be doing it? So we've had quite a few questions on that, and could you outline some of the plans?
So as a follow-on for the tofersen trial that we talked about, which is for the patients that have a SOD1 gene change, and that is 20% of patients who have a family history, but only 2% of the whole MND population. And so we identify patients who have that change, and sometimes members of the family also want to know if they carry that changed gene. So some people choose to, because if you have an affected mum or dad, there's a 50% chance of any one of their children carrying the changed copy of the SOD1 gene. Some people don't want to know whether they have that gene change, and that is, of course, their right. But some people wish to know if they have the gene change, even when they're fit and healthy. And we're doing a follow-on trial, again, Biogen are leading on this, called Atlas, which is, so we're taking those patients who have chosen to have that pre-symptomatic testing, and we're looking at them when they're fit and well, and then we're measuring neurofilament levels in the blood, a simple blood sample, each month. And there's a lot of evidence that as MND is beginning to develop, neurofilament levels go up. So if the person develops symptoms, or if their neurofilaments reach a certain threshold, then intervening with tofersen at the earliest stages. And we know from all the preclinical mouse studies that we've done in the past, the earlier you treat the condition, the better the outcome. So there's no doubt that trying to catch people early will be very helpful, but it's only for SOD1 at the moment. We're not doing that for the other gene changes that can be present in MND.
Okay, thank you. Rob, you're on mute.
Sorry about that. What an amateur, not an expert, you see. A huge amount to be positive about. When I think back to one of the last times I saw Doddy, he obviously he's 5.5 years into his disease and 5.5 years of leading the crusade to fundraise, to find solutions for it. And he delivered quite a depressing message in that he said for him as an end user, nothing has changed. There have been no developments. Obviously, he was fervently hoping that there would have been, but nothing had changed for him. But listening to what you're saying now, Pam, is that while for 98% of MND sufferers, there isn't any huge changes yet in terms of treatment, there's a huge amount coming on online. There's a lot to be hopeful about. And you were talking about the biomarkers. Is it worth explaining? You know, this is something that's come on a huge amount in the last 5 years in terms of finding biomarkers to measure the disease. For a layperson, what is a biomarker, and what's so wonderful about having worked out what they are?
So if I give you some examples, the reason that there have been such developments in diseases like various different types of cancer and multiple sclerosis is that there is something you can measure that tells you whether a treatment is working. So in cancer, you can measure the size of the tumor and see it shrinking if the treatment is working. In MS, you can measure inflammatory lesions using an MRI scan of the brain. And again, you can see a reduction in those if the treatment is working. We haven't had something we can measure, you know, either with imaging or with biochemistry in MND. We've had to rely on life expectancy, muscle strength, disability rating scales and so on. But the neurofilaments that I talked about, as motor neurons get injured and die, they release these neurofilament proteins. And what seems to be coming through from the recent trials is that if you've got a treatment that's working, certainly in tofersen, we could measure the lowering of the SOD1 protein, showing that the drug was hitting its target, and the lowering of the neurofilament proteins, which showed we were reducing the damage to motor neurons. And we could see those changes well before the patients reported improvement or stabilization of their muscle strength. So it's something you can measure. It might be imaging, it might be a blood test, it might be something in the spinal fluid that shows you that your treatment is doing some good. So really important, and they've led to huge developments in other areas of medicine. And now for the first time, we have that potentially in MND.
And am I right in saying, Pam, that one of the advantages is not only to find out if a drug is working, it's also to find out that a trial drug is not working. So you can take patients off the drug and not waste time.
Absolutely, it would enable you to have a go no go decision earlier. And I think the other exciting thing, we know, although MND appears as one thing in terms of the patient's symptoms, we know it's not one thing. There are various upstream causes. It's heterogeneous. And some of the trials we've done, because we didn't have a biomarker, we don't know within overall results of the trial that may be negative, maybe in there would be patient responder groups. So if we could identify a subgroup that got a lowering of neurofilaments, we could identify responder subgroups. And if you think about a disease like breast cancer, Herceptin, which is a relatively new treatment for breast cancer, if you just tried it in all the patients, you wouldn't get a very positive result. But if you targeted that treatment to the right group of patients, then it works really well. So I think that's the other thing, that having a biomarker like neurofilaments will enable us to pull out people who might get a good response and separate them from people who don't respond to that particular treatment.
Pam, could I also just ask about that? It seems to me that neurofilaments has become pretty established in a fairly short space of time in terms of being a reliable biomarker. Could it be used earlier for earlier diagnosis to try and identify the disease earlier? Because we do understand that the diagnostic pathway can be quite challenging and quite long for many patients.
Yes, and on average, it takes about 12 months for a patient from noticing the first thing wrong to get to the neurology clinic. So what we're doing with our hospital is setting up neurofilament measurements in a robust way as a clinical service within the hospital. And our dream is that, when a patient, if we can kind of highlight this to GPs, when a patient goes to a GP with my thumb is a bit funny, or my voice has changed a little bit, or I've got foot drop, then if the GP could measure neurofilament levels and they're elevated, they could be fast-tracked towards our clinic. So I think it might well enable earlier diagnosis.
Is there an expectation of other biomarkers as well, or is this the only one we need?
Well, I think it's fair to say it looks so robust from all the background work that's been done and from the Tofferson trial, but we need further evidence from more trials, I think, to really, so the regulatory agencies, the FDA in the US and the European Medicines Regulatory Agent, haven't yet accepted that. So we need evidence from more trials, but I think it really makes sense. And what we're trying to do is pull that through from the cell models, the mouse models. Can we, when we're testing a new therapy in those models, can we measure that the motor neuron injury is going down by the reduction of neurofilaments? And then we already know it's looking very promising in human beings. But I think we do need other biomarkers, but I think we've got a really solid foundation to move from now. In the commonest genetic cause of MND, which is caused by a mouthful of a gene called C9orf72. So about 10% of MND patients, 5 times as common as SOD1, about 10% of MND patients have a change in that gene. And there are other things that we can measure. So the change in the gene causes production of some abnormal proteins called dipeptide repeat proteins, DPRs, and we know we can measure them in the spinal fluid. So to a certain extent, your biomarker depends on what your drug does. So for the interleukin-2 trial, a good biomarker was the increase in Treg cell count in the peripheral blood, and we could also measure a reduction in a biomarker of inflammation. So I think there will be an array of them, but I think the fundamental thing about neurofilaments is they're telling us if we can reduce the level, we're reducing the injury to motor neurons. But there will be other biomarkers specific to a particular drug that's being tested.
So Pam, there's clearly, I'm also aware of the time. We were given a guidance of half an hour. We're already over that. So, but I just wanted to ask, in terms of the what seemingly is a plethora now of opportunities for clinical trials for patients, how would a patient go about insisting on getting on a clinical trial and being part of the whole research program?
So I think they really have to be assertive with their GP and their local neurologist. But I think the other very positive thing, we have for some time had a network of MND care centres, and we have a video conference every month to discuss trials. So there is a a network of centres. I think in MND Smart there are 17 centres around the UK participating. So it's not just the big research active centres that are recruiting patients to trials. One of our aims is to spread the expertise out so that all the centres can put their patients into trials, because you know, if you're doing an MND clinic, it's a very positive thing to be able to offer patients that we've got an experimental treatment that looks promising. You know, would you like to help us test it out? Most people are desperately keen to be able to try that. So part of our rolling program is to build up the confidence of centres that aren't so used to doing clinical trials and get their expertise at the right level. And I think MND Smart has been really excellent from that point of view.
That's great. Rob, do you have any final questions for Dame Pam? You're on mute again, Rob.
But you know, half an hour, and I'm still not an expert. In terms of what you were just talking about there, is you know, if we look at interleukin-2 potentially being something that almost all MND sufferers should be put on as you know, as a starting thing, would that be ruled out if people were going into a trial? Would they be not allowed, or you know, to be on any other treatments?
So that's a very good question, Rob. I think so no, we would, in testing an experimental treatment, what we try to do is put people onto best existing treatment. You know, at the moment, people are allowed to be on Riluzole and Edaravone, which isn't licensed in Europe, but is in the US. So you're allowed to be on best existing treatment, and hopefully that might include interleukin-2 before too long. And then on top of that, you add the experimental treatment or placebo. And for the duration of the experimental period, and then at the end of that, what we like to do, what we plan to do, is allow everyone to have access to the new drug. So best existing treatment, and then add on the new treatment.
Thank you.
Thank you. Sadly, we have run out of time. Thank you to certainly our two panelists. Pam and Rob, thank you very much for your questions and your answers. Quick reminder that any questions that haven't been answered, I think what we did, we grouped them into taking part in clinical trials, into biomarkers, and into people that carry genes. And those were the majority of the questions. There were plenty of other questions as well. We can't get through them all. So please do look at our website, and we will attempt to answer those in the best possible way. So thank you everyone for participating and watching our Ask the Expert, and your interest in our foundation. It means a lot to us. So keep fighting the fight, and thank you so much for your support, and good evening.
Bye-bye.
Thank you.